All right, let's talk about cells. So just some basics, as it says here. So there's basic structures that eukaryotic cells have. We'll focus on eukaryotes, which humans are, in this class. And we'll get a little bit into some organelles in this lecture that will mostly be in lab this week. We'll talk about some of the organelles. So let's get started here. A basic eukaryotic cell. Here it is. Pause the video and label some components of it. All right, we got the plasma membrane. So this is the cell membrane or plasma membrane that is actually a bilayer. So it's a phospholipid bilayer. Doesn't even quite look like this, but this is a little closer. We'll talk more about it next week. It's made up of the phospholipid bilayer, you know about phospholipids, as well as some proteins in there that are going to be really important, and some cholesterol. The purpose of this membrane, the structure, is to separate the extracellular fluid, or ECF, from the intracellular fluid, or ECF. ECF is outside the cell, ICF is inside the cell, and ICF is also the cytosol. So it's actually the fluid that's inside the cell. And we'll talk more about those two, the components of each of those. They're really important in terms of the ion concentrations, for example. But the membrane is important because it separates them, right? So it's going to be able to keep that inside of the cell a certain way. So maintain homeostasis inside the cell in terms of ion concentrations, everything the cell needs to have to function. So it's one of our barriers. Then we've got our nucleus. That's where the genetic material is stored inside of a eukaryotic cell. So DNA in the case of a eukaryote. So it contains genetic material, a few exceptions, but most cells, eukaryotic cells, have a nucleus. Then we've got the cytoplasm. Cytoplasm is everything outside of the nucleus. Very broad term. It encompasses both the cytosol. What's another word for cytosol? ICF. The cytosol is actually the aqueous solution. And then the organelles. So cytoplasm equals cytosol plus organelles. So cytoplasm is a lot of components to it. This is the basic. The complex is this, right? Here are our organelles. So first of all, we've got the same structures, our plasma membrane around here, our nucleus, the DNA is packaged into chromatin. We've got our nucleolus as well. Then we've got tons of organelles and this really dense, all these dense structures. So endoplasmic reticulum, the rough ER where proteins are made. We've got ribosomes floating around in here that actually do the protein synthesis. We've got mitochondria, an important one for production of ATP. And we will talk about some of these in lab. We won't actually see most of these structures in lab. So when you see if you even see any cell components in lab, it'll be the nucleus, cell membrane. It'll look a little bit more like this. So this is what you can see with a light microscope in terms of a view of a cell. This would actually be probably a multi-lobed cell here, multi-lobe nucleus of a white blood cell. And that's about all you can see. There's probably some lysosomes that you can see around here as well. And the reason we can't see better than this is because of how small cells are. I want to go to this website where you can see, it's a really nice resource for kind of getting an idea of size and scale of molecules. So when we're looking under the microscope throughout the semester and year, it's good to have some idea of what you're looking at, how big it is, etc. So here is a coffee bean. You can actually go to this website yourself and scroll in yourself, but it gives you kind of an conceptual idea. Grain of rice, salts, I'm sorry, you know how big that is. You can barely see it before we get down into what you can't see with the human eye anymore. Human egg is a single cell, so is a paramecium. Very big cells. Then we got a skin cell. Skin cells are something you will see in lab, so be able to kind of get the idea of the size of that. Red blood cells are also eukaryotic cells, a lot smaller than skin cells. So there's diversity even in the human from this size to this size. let's go down to mitochondria is there. So the nucleus would be bigger than this. It might be about the size of yeast. And that's about what you'll see inside of the cells we're looking at. There'll be some cilia, another structure you'll see that are a little bit smaller. We can go even farther just to get an idea of, in mitochondria, there's viruses in here, components of viruses, ribosomes. Hemoglobin is a protein, right? There's our hemoglobin protein, glucose, 6 carbons, phospholipids were really small now, amino acids. These are biomolecules that are inside of a single cell. So I encourage you to explore this to kind of get an idea of size and scale. Your book has a nice description, nice pictures as well. Last thing about cells for now is the various cell shapes. So along with the different proteins that are produced due to cell specialization, this results in different cell shapes. So I can introduce them here. This is related to our differential gene expression, and also we're going to be seeing a lot of these types in lab in terms of the different tissues we're looking at. So some of these are types we'll see in epithelial cells, so squamous, cuboidal, and columnar you'll be seeing in our first microscopy lab as well as with the tissue lab. So these are different shapes that make the size look a little different, right? Even though they're all, they probably have about the same volume, these ones are probably a little bigger. Discoidal, those are primarily hemoglobins. Those are typically going to be a little smaller, but we've got all these shapes we're going to see. So epithelial cells in the skin will look like this. Fusiform is going to be our spindle-shaped muscle cells often. And then fibrous are going to be a lot of our keratin, collagen, other parts of connective tissue. So just be aware, right? Just like this is type of cell specialization, they can come in all different shapes as well. That's the basics of cells. Here are the 2 learning outcomes. Just introduce you, identify and describe the various cell shapes. That was the second one we did. Got the list there. And then identify and describe and draw the main parts of a eukaryotic cell. And I'm realizing I didn't actually talk about all of these here, so I'm actually going to go back and do that quickly now.